Okay, so in this video, I'm going to show you how to use Matlab to construct single-cell gene regulatory network. So I'm loading a new example data, then I start to use scatter plot to visualize it. So you can rotate your cells to look at a different population of cells. To use all cells to construct a single-cell gene regulatory network, you can click this button here. And then there's two ways. You can either select a list of genes or paste a list of genes. In this first, I'm going to select. And here we have more than 9,000 of genes. So this is a long list. You can select some of genes as an example. And I'm going to just randomly select a few genes to play with. And then once you finish, click done. And then the network is constructed using an algorithm called PC regression, principal component regression. And you can see this network is directed and weighted. And also there's positive and negative signed. The blue linker means the positive correlation between genes, and the red link indicate the negative correlation. See, if I click this button, I will change directed into undirected. I can switch, I can apply weight, so I have some ideas, the different weights of the edges. And I can select cutoff. So if I use 30%, most of the links will be retained. If I use 85% cutoff, then only 25%, 15% of the edges will be left on the plot. And I can change the size of the gene name, and again different cutoffs, and I can change the layout. Alright, at the end I can save the adjacency matrix to the workspace and also the list of genes. So that's the construction of single-cell gene regulatory network using all the cells. Now next question is, we are going to compare networks constructed from different cell populations. In this examples, I have 5 different groups of cells, and the blue means the group 1, and 2, group 2, and 3, yellow cell group 3. And I'm going to compare gene regulatory networks constructed from a cell in population 1 against 3. In that case, I have to press this button, and then I will select based on the class ID. I will compare class 1 against 3, and then in this case I'm not going to select any gene. I just paste a list of genes. In this case I just randomly select a couple of genes, and then 2 networks will be constructed. Again, you can see changes to the unidirectional, and I select the weight. So let me show you the weight first. Highlight the weight of the gene, of the edges, and then you select 80% or 90% of removed. And then to compare these two graphs, you will be able to identify some varying events. For example, ASL gene wasn't connected with many other genes, but in sample 2, it has a lot of connections with other genes. And there's another automation cutoff you can select to show the progress of cutoff of different settings of cutoff apply to pull the edges gradually from the network. Through this, you can have a visual detection of the change of patterns in network connectivities between these two networks. All right, that's the end of the demonstration. I hope you enjoyed. Thank you.